Hello, I'm Dr. Gor Sarkisian. This is Biochemistry 2. Last lecture, we started talking about certain phosphorylated compounds with large and negative delta Gs that were other than ATP. And today we will continue on that topic and we will begin today's lecture with discussing thioesters. Well, first of all, what is a thioester? It is a molecule that has a sulfur in place of the usual oxygen in an ester bond. Okay? The hydrolysis of this bond is associated with very large and negative delta G. There are many common biologically relevant thioesters. One that you're very well familiar with is acetyl-CoA. It has acyl group that is basically ready for transacylation, condensation, or oxidation reduction reactions. Transacylation, what does that mean? It basically means a transfer of the acyl group from one molecule to another. So let's see what that looks like. Here we have a sequence of events. We begin with our acetyl-CoA. Here we have our ester bond, thioester. We hydrolyze this bond, obviously with use of water, in which we have an H group being taken away with CoA sulfur H bond. We add basically this hydrogen to the sulfur right here. And then we are left with acetic acid. Now, what is interesting in the aqueous solution is that this acetic acid can be ionized. Basically, this hydrogen ion can be released, and with that we generate this partial negative but diffused charge between these two oxygens. Okay, and this reaction is spontaneous and it's reversible. Of course, it drives towards the acetate formation, but it's reversible. Moreover, the hydrolysis of acetyl-CoA, as you can see, that creates acetate in the end, and CoA plus hydrogen ion, it has a overall delta G of minus 31.4 kilojoules per mole. Hydrolysis of thioesters generally yields larger delta G than their oxygen-containing ester counterparts, because the oxygen esters can have a resonance stabilization in which basically this double bond can be diffused between both of these oxygens, and we have a diffused negative charge throughout the ester region. Okay, so hydrolysis, in this case, hydrolysis of thioester and oxygen ester, in both cases we generate carboxylic acid, the same product. Well, in this case, this carboxylic acid is an acetic acid, but the point is this transition versus this transition there is a net difference in delta G between these two um, hydrolysis reactions. So throughout this course, we have seen the following expression: ATP plus H2O produces ADP, an inorganic phosphate. This is simple to see and understand that this is a hydrolysis of ATP, but how does this reaction drive chemical processes forward? Well, in reality, the ATP provides energy by actually transferring a group rather than a simple hydrolysis of itself. Now, transferring a group, what group might that be? Let's see. So here we have a diagram, and it shows the chemical reaction that produces glutamine from glutamate. And of course, this reaction is as such energy unfavorable, therefore we need to have the help of ATP to drive this reaction forward. And we have seen this previously, and we accept it as it's a ATP mediated reaction. So by simply looking at this, one might assume that it is the hydrolysis of ATP itself that drives this chemical reaction, which is, needless to say, well, wrong. Just the hydrolysis would generate heat. And as such, as we have talked about in a previous lecture, if it's simply heat, we cannot use that heat to perform any work because biological systems are isothermal systems. So in reality, what happens is the following. We do have the transition of ATP to ADP, which of course means a loss of a phosphate, adenosine triphosphate to adenosine diphosphate. Now, what happens to that extra phosphate? Well, because we have a glutamate here, and glutamate right here has a negative charge, we basically transfer that phosphate onto the glutamate, the ester bond here, making an ester bond. So this is a glutamyl phosphate. Now, this glutamyl phosphate is a very high energy molecule. It is enzyme bound, and if you remember from the last course, sometimes high energy molecules can be in a stable form within the catalytic site of an enzyme. So only after this intermediate high energy molecule we have actually the ammonia actually coming in a chemical or creating a chemical bond with the ester to form a glutamine, which is the step in which the inorganic phosphate is again liberated. And of course, in the end, we do get ADP, which is right here, and the phosphate right there. On a small scale, however, some processes do involve just direct hydrolysis of ATP or GTP. Remember, guanosine triphosphate. For example, a non-covalent binding of ATP followed by its hydrolysis, or GTP, followed by its hydrolysis um, to its constituents of ADP and phosphates, shall we say, can provide the energy to cycle some proteins between conformation. Now, it's not a chemical reaction. It's basically going from one conformation of a protein, let's say, to another. It can also, well, as we will see later on in this lecture, such conformation changes could be very important in muscle movement. Okay, but it can also help in movement of enzymes along DNA and ribosomes along RNA, and it can also, such hydrolysis can aid in reactions catalyzed by helicase, REC-A proteins, and some topoisomerase. So, helicase, REC-A, and topoisomerase. So here's a question that please answer and send to me as an assignment. What are these? Give a brief description, where they are, and what they do. Why are they important? In biological systems, the phosphate-containing compounds are generally categorized as high-energy compounds having large delta G, which is also negative, and we have low-energy compounds having less negative delta G. This is a little bit, well, arbitrary, but for example, the hydrolysis of ATP, we know is it has a negative delta G of minus 30.5 kilojoules per mole, or hydrolysis of glucose 6-phosphate has a delta G which is still negative, but it's only 13.8 kilojoules per mole. So the glucose 6-phosphate, therefore, is considered to be low-energy phosphate-containing compounds. Now let's look at how we can make use of this distinction. For ease of use, high-energy phosphate compounds, well, ATP can be considered a high-energy phosphate-containing compound. However, on this scale, you can see that we have some compounds that are of higher energy than ATP, like phosphocreatine, for example, the 1,3-bisphosphoglycerate, and the phosphoenolpyruvate, or otherwise known as PEP. So now let's look at a coupling of these reactions. The hydrolysis of PEP plus H2O yields pyruvate, right? Plus an inorganic phosphate with overall delta G being minus 61.9 kilojoules per mole. Okay, now our dehydration of ATP or ADP. ADP plus phosphate, that should yield adenosine triphosphate plus H2O. This reaction has a delta G of, obviously, 30.5 kilojoule per mole. So this reaction is not happening just by itself. Okay, now let's open up a new page and see what's actually happening there. So even though the delta G of the final sum reaction is the algebraic sum of the two, the final reaction is basically a standalone reaction in which we assume that PEP directly transfer a phosphate to ADP, and then we produce pyruvate and ATP. Now let's look back at our previous diagram. We have what's called high energy compounds again. We have low energy compounds, and glucose 6-phosphate in this case is a low energy compound. We have very high energy compound as PEP, and we have something in the middle which is still a high energy compound, but it is somewhere in between these metabolic molecules and the low energy molecules. Okay, so therefore ATP finds itself energetically in a very favorable position between these high energy and low energy compounds to serve as a universal energy carrier in all living things. And it just happened that all living things have found ATP to be this golden standard in energy transfer. Another equally or maybe even more important characteristic of ATP is that even though it has high energy, it is for practical purposes kinetically stable, which is actually good news. So it doesn't just decompose in an unpredictable manner in the cells. It does not spontaneously phosphorylate other groups or give the phosphate to in an uncontrolled manner. And furthermore, these transitions of phosphate can happen in very specific way, in very specific enzymes, the concentrations of which the cell can control. Therefore, the cell can regulate the rate of hydrolysis of ATP and its phosphorylation potential. Now let's look at what ATP can produce or do in terms of chemistry. Well, it can produce a phosphoryl group and ADP. It can produce pyrophosphoryl group and AMP. Also, it can produce adenylyl group and pyrophosphoryl. So the difference here is that we are talking about a transfer. So transfer of a single phosphoryl group would be phosphorylation, and transfer of adenylyl is known as adenylation, and we will talk about this in a few slides. So any of these phosphates, alpha, beta, and gamma, are subject to SN2 substitution. And from basic chemistry, you must remember that SN2 is a kind of a nucleophilic substitution mechanism in which basically one bond is broken while at the same time another one is made, obviously. Now let's chop this down and look at it one by one. And we will begin by looking at the gamma phosphate first. So what happens is that it basically displaces the adenosine diphosphate, and it creates a new phosphoester bond. So if we have or conduct an experiment in which we use a radioactive oxygen, we can actually trace and see if it is actually this oxygen that is being released, or is it this? Meaning, do we have the oxygen that is being reacted, the nucleophile, is it actually connecting to ADP, or is it actually connecting to the phosphate? And these reactions show that we do have the nucleophile actually reacting with the phosphate. So the phosphoryl group in this case will be transferred onto the compound that has this or performs this nucleophilic attack. In the same way, the nucleophilic attack onto the beta phosphate results in pyrophosphoryl group generation, and again, the nucleophile is actually being connected to the pyrophosphate. Therefore, we actually transfer the pyrophosphate from ATP onto the nucleophile. And finally, if we have a nucleophilic attack on alpha phosphate, we actually get the nucleophile being attached to the adenylyl portion of the ATP. So we transfer adenylyl group here, and that process is known as adenylation. So again, we talked about this, but I'm going to mention it one more time. Whenever we have an attack, a nucleophilic attack on alpha beta, we have much larger release of energy because we have the pyrophosphoryl being generated, and the alpha beta is about 46 kilojoules per mole, whereas the beta gamma is the standard 31 kilojoules minus. So, but besides having large 46, the minus 46 kilojoules per mole, the generation of pyrophosphoryl can undergo further decomposition into inorganic phosphate and release additional 19 kilojoules per mole of energy. Now, the one important side effect of this, I suppose, is that the 19 kilojoules per mole that is released from this pyrophosphoryl breakdown to inorganic phosphate adds extra energy for adenylation, because we have the adenyl right here. So it pushes or promotes the adenylation reactions also. Adenylation reactions, therefore, are very, very um, energetically favorable reactions. A good example of adenylation is the activation of fatty acids, for example. So, and in reality, when the ATP is used to drive particular unfavorable metabolic reactions, adenylation is often a mechanism of energy coupling. I would like to briefly show what um, adenylate can do, and a good model here is the firefly, because the cycle of generating light by fireflies is as follows. We have the primary molecule that generates the light of firefly is luciferin. And when luciferin is coupled with adenylyl, well, adenine monophosphate, right? Adenylyl. This molecule is primed to perform work. It's high. It's in at a high energy state. So it can actually undergo oxidation through with the help of an enzyme called luciferase, which emits light and produces carbon dioxide and adenosine monophosphate. And it produces the rest of the luciferyl adenylate molecule, which is oxyluciferin. Oxyluciferin then can be recycled to luciferin, and importantly, we have a negative sign here, charge. And then through hydrolysis of ATP and transfer of basically the adenosine monophosphate to the luciferin, we restore our luciferyl the adenylate, luciferyl adenylate, which then can undergo the same chemical reactions and generate more light. So we form our good old ester bond here between the luciferin and adenosine monophosphate. Now let's look at what else can ATP do. It can create electrochemical gradients. It can result in muscle contractions. Now let's see how. ATP can, for example, support the transport of ions where its concentration is high against its concentration gradients. For example, in kidneys and in the brain, a large amount of the energy is consumed to pump sodium and potassium across the plasma membrane by a sodium-potassium channel, which is an ATPase. ATPase, a molecule that hydrolyzes ATP. Now, how does this work? Well, it's a cyclical phosphorylation and dephosphorylation event. And interestingly, the phosphorylation event is coupled with sodium, which, of course, a protein is phosphorylated and it changes its conformation. And the dephosphorylation of this protein, of this transporter, sodium-potassium transporter, is the dephosphorylation is coupled with potassium, which obviously dephosphorylated results that it goes back into its native conformation. Each cycle of this phosphorylation, dephosphorylation, of course, converts an ATP to ADP and a phosphate, and that results in electrogenic pumping of sodium and potassium. Now, here's a question. What is electrogenic pumping? Another way that ATP can support work to be done in biological systems without using the potential energy of heat or transfer of phosphate is in the in a skeletal muscular system. Well, not only skeletal muscle, the smooth muscular system also does the same thing. But in either case, we have actin and myosin, which are specialized to use the chemical energy from ATP to convert into motion. ATP can bind to myosin non-covalently, but quite tightly, and tight enough for myosin to hold a particular conformation when ATP is bound to it. So myosin then can catalyze the hydrolysis of ATP that is bound to by an enzyme called myosin ATPase, therefore generating the ADP and phosphate. But the point is that it results in a release of ATP from myosin that in turn results in relaxation of the protein into the second conformation, up until the next ATP molecule binds to the myosin, and we see this process, this circular process occurring over and over again. So there's this micro changes in conformation of myosin result in overall movement of the muscle. Remember, we always talked about myosin walking over actin. So let's say this is going to be a very crude and maybe even funny drawing here. But let's say this is one leg, this is the second leg. When ATP is not bound to it, it's in this conformation. When ATP binds to it, ATP is bound, then it's in this conformation. Then when ATP releases, it goes back to the original conformation. So therefore, it forms this walking type of behavior. Funny, but this is exactly what's happening in interaction between actin and myosin and the hydrolysis of ATP. So we've been talking about ATP, and as it is the main energy transport machinery in our organism. And it is true, it is the case, but besides ATP, we have, of course, other triphosphates such as GDP, UTP, CTP. And energy-wise, they are about the same as ATP. Same thing holds true for deoxynucleotides, triphosphates like DATP, DGDP, and so on. They are energetically comparable to that of ATP. Now, these various nucleotides are involved in vast majority, many different biological functions. Well, DNA, for example, and these are generated and maintained as a nucleotide, as nucleotide triphosphate, and NTP, right? Nucleotide triphosphate. In this form by phosphoryl group transfer to the corresponding nucleoside diphosphate. So obviously nucleoside diphosphate, right? And also the nucleoside monophosphate, just like ATP, adenosine, guanine, cytosine triphosphate. So basically, the reaction that generates the nucleotide triphosphates are almost the same as anything else, which is ATP plus nucleotide diphosphate, or of course, dinucleotide diphosphate, or dioxynucleotide. And of course, We did talk about this already. It's in the presence of magnesium ion. And it, of course, yields ADP and yields our nucleotide triphosphate. And this reaction is catalyzed by nucleoside diphosphate kinase and the nucleotide diphosphate kinase. Now, this reaction, which is catalyzed by nucleoside diphosphate kinase, is fully reversible, and that relationship is shown by equal length arrows between these reactions. It is the relative ATP to ADP ratio in the cells that normally drives this reaction forward. And we basically form the NTPs and DNTPs. So it's basically the concentration that drives this. So now let's look at the mechanism. It's quite fascinating. We have, of course, it's a catalytic reaction in which we have adenosine triphosphate releasing one of the phosphates to an enzyme that has a histidine residue. This generates an ATP, ADP, and phosphorylated enzyme. And then this phosphorylated enzyme can release this phosphate to nucleoside diphosphate, forming the nucleoside triphosphates, which are ready to go do their job, like being incorporated into DNA. So an interesting phenomenon is that if we have a lot of the ATP being hydrolyzed to form ADP and phosphate, and then the phosphate is removed through some other reaction to, let's say, as we just talked about, to make NTP, then we have an accumulation of ADP. And the more ADP we have in a surrounding, that's our product of our reaction, that will slow down the hydrolysis, right? After everything is said and done, a reaction is still a function of a concentration of product over reactants. So, for example, when a muscle is in a vigorous state of exercise, we have accumulation of adenosine diphosphate because, as we just said, we have our hydrolysis of ATP drives the actin and myosin interactions, right? So, well, this accumulation of ADP in the muscle, well, it's not good. It's not good. So muscles have to be able to accommodate for this accumulation of ADP, and luckily we have an enzyme called adenylate kinase. Now, what's adenylate kinase? What does it do? Well, it basically takes two ADPs in the presence of course, magnesium, and it generates an ATP and adenosine monophosphate. Now, the delta G of this reaction is a whopping 0 also. So this adenylate kinase is doing two jobs, or is killing two birds at once. It reduces the concentration of ADP and it produces an extra ATP for the muscle to use. Another very useful reaction is this, which involves phosphocreatine. This also, well, phosphocreatine, it also creates phosphate. It's a good reserve for the phosphoryl group for quick synthesis of ATP from ADP. So promote this reaction, basically. So the concentration of phosphocreatine in skeletal muscles is about 30 millimolar. That's about 10 times the concentration of ATP. Okay, and same thing holds true for smooth muscles and kidney and brain. In those organs, it's 5 to 10 millimolar. So this reaction is catalyzed by creatine kinase. So it's creatine kinase. So when a sudden need of ATP arises, the phosphocreatine reserve could be tapped into to generate ATP very rapidly. So with this, I believe we will conclude today's lecture. We will start talking a little bit about inorganic polyphosphates, and we will go on to the next segment of this chapter, and we will start talking about the biological oxidation reduction reactions. Thank you. I shall wait for the few questions that I asked in the presentation. There were only about 3 or 4 of them. I will post if anything else is needed, I will post them online. Thank you.